anybody who shows up in a few minutes. So I'm Terri Gilbert. I'm from the Allen Institute for Brain Science. I'm the application scientist, which means I interact with all of our users, training people how to use our data sets and interacting with anybody who has questions. So if you have any questions, one of the ways to get a hold of me is from my email address here. You can also contact me on Twitter. Sometimes that's faster. So, but how many of you are familiar with the Allen Institute, first of all? Excellent. So the other question I want to find out is how many of you are biologists or neuroscientists? How many of you have no biology, don't know biology very well? Anybody? Okay, good. So because Ariel let me know that there's a couple things. So there's a couple basic slides that I put in there just so that you can get a sense of the biology behind what we're doing. But so I'm all kinds of assumptions about you people. Yeah. Okay, so I'm going to start with this slide, which people usually finish with, because I usually finish my lectures all over the interwebs. So this is the group of people. It's not everybody who have been involved in the work that we do. So this is taken at the Allen Institute. And again, I can't say enough how much gratitude we have for Paul Allen. I love what he's doing with his money. So it works for me, and hopefully it works for you as well. So part of this, the intention of this is to let you know of the resources that are available for you. So you might know about it already, but I want to give you a sense of everything that's available. Some of it won't be of interest to you right now. It may be of interest to you later. So I'm letting you know so that you can at least come back later on. So I'm going to give you a little bit of a background of the Allen Institute and some of the methods behind some of the work that we're doing. I want to show you the data that's available in the Human Brain Atlas so that you can use it intelligently. There's one of the best things about finding out what data is actually included is what not to do with it. So I'll show you how to do that. I'll show you how to have access to the data, and then I'm also going to talk a little bit about some of the resources that other people have created based on our data, so that you have access to that as well. And then I'll go live and I'll show you the portal, and then that part is mostly going to be based on what it is you're interested in seeing. But I want to let you see our website, the web portal, as well as how to access the data, all of the data, all of the data that we have. So one of the ways I like to interact with people is via poll. So this is one of the, you can participate right now. Those of you on Twitter, you can respond by just tweeting the answer to this question. The first question is very simple. What is your first name? You can also go online and respond here, or you can text. But this way, this gives me an idea of what might be of interest to people. There we go. And I also like capturing, especially this image, just to gets a sense of the people that have participated. And then once you've participated in this particular question, you're set up for any of the following questions that come up. I was about to say, I know there's a couple Chris's, yeah. Oh, I like that with the smiley face inside the H. That works really well.
Wait, why is this so big?
Because there's two Chris's. Two people have entered.
Like 5 Anishas.
People tried to game the system that way, so I set it up so you can only respond once.
I see that.
Yes. Don't. Okay, great. I love this.
I'm going to interrupt you.
Yes.
When you went to our
Okay, great. But so now you're set up for participation later. Okay, I want to talk a little bit about where the idea for the Allen Institute came from.
The brain is a never-ending source of fascination for me. It's the organ that unites us as a species and distinguishes us from one another. In March 2002, I invited 21 scientists, including 4 Nobel laureates, to join me at a 3-day brainstorming session. By the second day, the conversation kept circling back. What neuroscience needed more than anything else was something very basic. A better map of the brain.
So that's just a short version of that particular video, but we just sequenced the human genome. So we knew what genes were available. We knew that, you know, the proteins that genes made were in the brain. We just didn't know where they were. And so the idea was based on this very central dogma, which is that DNA makes RNA, makes protein. And while the protein often is what does the very interesting things, it's hard to scale that up to map where proteins are in the brain. But it's relatively simple to look and see which genes are expressed where. So the idea of mapping gene expression in the brain is where what they landed on at that meeting 15 years ago. So I want to talk a little bit about a couple ways to look at this. So colorimetric in situ hybridization is what a lot of our atlases were first based on, especially back in the day when we first started this work. So you have slides, pieces of brain that are on a slide. These are, this is actually slides taken from our data set. But each of these slides, in each one of the cells, there's RNA just floating around. And you can actually create a probe for a specific piece of RNA. And in this case, we've labeled the probe so that we have antibodies that bind to that probe. And then there's a couple of amplification steps. And what results is a purple precipitate in those cells in which the gene is expressed. So this gives us a map of where that particular gene is expressed. And you do a single gene across the entire brain, and we did that for 20,000 genes. And we did it primarily in the sagittal plane, but we also did some of the genes in the coronal plane as well. So this is a rather long process anyway, and we were able to scale it up and map all the genes, the gene expression in the mouse brain in 3 years, all 20,000 genes. So, and then there's a lot of really cool informatics that went on top of that. Any of you familiar with the mouse brain gene expression atlas? Okay, great. And so we sliced up the human brain, or the mouse brain. This is the mouse brain. 20,000 genes in the sagittal, 4,000 genes in the coronal. There's some really cool web visualizations that utilize just the coronal genes. And this is what it looks like. It's hundreds and thousands of images, often where you've got gene expression in a very discrete area. And you'll also notice that there's a lot of background, which human beings don't like so much, but computers love. And then we also did this in the developing mouse as well. So this is genome-wide. The developing mouse brain atlas is not genome-wide. It's only 2,000 genes, but it's got as many images in it as the original atlas because we looked across 7 developmental stages. So, but when you want to do this in human, there's a couple things that make it really hard to look at gene expression in human the same way. So first of all, this is the size of a mouse brain compared to the size of a human brain. And we used about 3,000 mice to capture all 20,000 genes in the mouse brain. You can't do that with human. First of all, we'd still be slicing up a human brain trying to get the gene expression in the human brain. But also these mice were C57 black. They were a highly inbred strain, so the assumption was that all of the brains are identical, and you just can't do that in humans. So we had to take another tact when looking at gene expression in the human brain. So what we used was microarray analysis, and in this case what we did is we we took out a sample of the brain from a particular area of the brain, and then we mapped that back into the MRI from where we took it. And then we extracted the RNA. We, using reverse transcriptase, created complementary DNA with a fluorescent label on it, and then you run that on a chip, right? And then what then results, this is actually an image of one of those chips. We used 60,000 different probes looking at all, yeah?
How big physically were the tissue samples?
About a millimeter by millimeter by millimeter. Yeah, just a tiny little chunk across all 6 layers of the cortex when we looked at it in the cortex. And I'll talk a little bit more about the data.
Each probe had the same size, roughly.
Yeah, roughly. And there's a lot of normalization that goes.
Well, you call them wells.
Yeah, well, right. You've obviously looked at the data, so yes. So, but yeah, all of that data is in the documentation. So, one of the things about our data is it's open source. It is, publicly available. You are welcome to publish on it. In fact, people do. And later, Kirsty, one of your own, is going to be talking about her. She was on a paper 2 weeks ago, PNAS paper, where she'll describe how she accessed the data and then what she did with it. So those are 2 different kinds of methods that we use to pull out gene expression, and we did in situ hybridization also in the humans, but it's not like we did in the mouse. So it's a limited number of genes and a limited number of structures. So, but what I want to talk about is where we came from and now where we're going. So some of you might not be aware of what we're currently doing at the Allen Institute. So based on all the genetic tools, we did set out to map the gene expression in the brain, and we pretty much accomplished it. But it's not comprehensive, it's not exhaustive, absolutely not. But it gave us enough information for us to be able to start asking what we think are more interesting questions, like how does the brain work? But we based a lot of it was all based on the work that we did with the gene expression, and we started to create genetic tools in which we can ask other kinds of questions now. So how many of you are familiar now with the work that we've been doing at the Allen Institute? Anybody familiar with our recent work? Okay, good. So I'm going to talk a little bit about that. So we're interested in the connectivity, and then we actually are interested in cell types work as well as the molecular profile. So we're still doing gene expression, but the gene expression that we're doing now is at the single cell level. So we've got a anatomical mesoscale connectivity atlas. Anybody familiar with the connectivity atlas? I'll just mention it. I'm not even going to talk about it. But if you're interested in it, there's a lot of really cool data in that data set as well. And then we've also got our cell types database where we've patched individual cells, looked at the recordings, filled those cells. So some of those cells actually have morphology to them. And then we've also, in a separate study, have single cell RNA-seq. So that data is available in the mouse currently. And then we're also doing full morphology in the brain. So we've actually, done single cell recordings in a live brain, and then filled that single neuron and to see where it projects in the brain. And that data's not online yet, hence the pink. But once we can put that data online, that's also data that you'll have access to. And it's fascinating to see where a single neuron, as opposed to a population of neurons, projects to. And then. So we want to have refined mesoscale and microscale connectivity. So we're looking at electron microscope, using the electron microscope down at the synapse level, as well as array tomography. And what we'd like to do, and what we're starting to look at now, is measuring single cell RNA-seq in cells that we've already done an electrical recording from, so that we can connect all of that. It's what we call being able to correspondence problem, being able to see all the way through gene expression, through the electrophysiology, morphology as well. And then the synaptic physiology, that's our array tomography. And what we're interested in doing is understanding the connectional properties as well as the single cell properties, and we're also starting to do behavior as well. So having mice doing a visual behavioral task, and to be able to map that all the way across all of the different brains. And one of the things that we've gotten really good at doing is once we have all this data, yes?
To clarify, this is for the mouse brain?
Yes, and some human. So,
Which part is here?
we're working with several doctors in Seattle where pieces of cortex have been resected for either epileptic or cancer, and then we get the cortex.
Physiology is also.
The electrophysiology, yeah. And there's, we're one of the things that we're doing is we're also creating a common coordinate framework for humans, like we have for our mouse, so that we could, at some point, this is a plan for the future, be able to map into the brain those particular kinds of neurons, right? Right now we're also interested in seeing how different from a human or from a mouse, besides just the size, is a human. So that we've started to collect some of that data, and that data is going to go online next year. The electrophysiology and the morphology. Yeah.
Is there any AIBS for humans?
We are also doing that as well. So that's we're doing the nuclei right now, and we're getting some decent results. But that data won't be out online until next year as well. Okay. So this is a timeline of the different products that are available for you to use. So we started in 2004 with the Mouse Brain Atlas. It's an in situ hybridization atlas, genome-wide. We also have done the Spinal Cord Atlas. It also is genome-wide. It's in 2 developmental stages, P4 and P56. And the Developing Mouse Brain Atlas, which is also in situ hybridization. Again, it's only 2,000 genes, but it's across 7 developmental stages. And then, in particular, these 2 have unique informatics visualization on top of the data that we've collected, so that you can do things like query the data itself. You can actually look for genes that show a high expression, or look for genes that show a similar expression pattern. We've also always been interested in the Human Brain Atlas, so that's, I'm going to be talking about the Human Brain Atlas today. It turns out where you can't control for humans, you can control in primates. So we've actually got some really decent data, both in situ hybridization as well as microarray data in non-human primates. We've also got a Developing Human Brain Atlas. So again, it's not, you get what you get in this one, but we've got donors from 8 post-conception weeks all the way up to 40 years. That's RNA-seq data, and Jeremy is going to be here from the Institute talking about that data more tomorrow. So this is at the point where Christoph Koch joined the Institute, and he's, I don't know if any of you know who he is, but he's interested in finding the neural correlates of consciousness and understanding, how the structure of the brain actually produces all the cool stuff that the brain does. And so that's, our 10-year plan back in, we created it in 2012. And that's where we've started to work on what I say the more interesting questions, as opposed to just mapping where the brain is, or where gene expression in the brain is. So we have our Connectivity Atlas. Again, if you want any information on any of these other things, I'll be around for the next couple days, so you can come ask me. Our Cell Types database and our Brain Observatory are our latest data sets that are incomplete at this point. We're still putting data into it. And one of the things about the Allen Institute data is, one of the things we do is we collect a bunch of data and then we release it online. And then we start to go to work on the interesting informatics to put on top of it. So these data sets, we just don't have enough data yet. I think we have about 800 cells, ePhys as well as morphology. There's only about 200 cells in morphology. We have 18,000 cells in the Brain Observatory. And the Brain Observatory is calcium signaling in the cortex during a visual stimulus. So there's several different visual stimuli, and we looked at the change in fluorescence due to activation it's 4 different areas of the cortex, of the visual cortex. So this data is our newest data set. It's our rawest data set, but it's easily accessible through the SDK, which is our Python wrapper around the API. So we also most of the stuff that we do is control brains. So our humans control, right? You know, how do you find a normal human? Exactly. But, you know, we work on not having any diseased brains, except for we do have a couple data sets that you may be of interest. So there's our aging, dementia, and TBI data set, which is mild TBI. It's not CTE grade concussion, but it is mild TBI. And the interesting thing about this data set is we don't find a whole lot of we didn't find a whole lot of correlation between dementia and TBI, and it, like I said, mild TBI, which is mostly how that affects us, right? You know, I actually had a traumatic brain injury and loss of consciousness when I was 7. It turns out that I have now 3.5 times greater chance of developing Parkinson's disease, but not dementia. So. So this data set, as well as the IV glioblastoma data set, are things that are, there's a lot of human data that you may actually be interested in, and Jeremy will talk about that tomorrow. You had a question?
And the electrophys
Yeah.
coming from epilepsy and tumor.
It's the normal part of the cortex that has to be excised to get at either the foci or the cancer. So, not to say that's normal brain, but it's the kind of human brain that we have access to.
It's the best you can get.
Exactly. So it's the normal-ish part of the diseased brain. Yes. So
as far as we know.
As far as we know, exactly, and hopefully we'll know more as we collect that data.
Don't get me wrong, this is the best we can do.
Right, exactly. You know, people don't like donating their brains while they're using them for some reason. So, any questions about anything here? So there's microarray data in the Human Brain Atlas. There's also some in the Developing Human Brain Atlas that you have access to that I'm going to talk about. There's also a small data set in the Mouse Brain Atlas. So if you're interested in that, I can show you how to access that. There's one of the things about my training people on these data sets is there's so much stuff to show you. I can't possibly show it to you in a day, much less an hour. So any questions anybody might have about anything so far? Okay. And then tomorrow, Jeremy's going to be talking about the RNA-seq in these data sets. So as well as some of the other data, if you're interested in that. There's a lot of high-dimensional data now that we're starting to collect. And okay, so here I'd actually like the answer to this question to see if there's resources that you're probably going to be interested in, if not this week, then later on. So, no, actually in this one. I think you can select more.
So while we wait for
Yeah.
the question, so I heard that you have stored additional donor brains from human cadavers.
In the freezer. Yeah.
That are just waiting for revival of the project.
Right, yeah.
So any news on that? Is that high on the priority list?
Well, I'm not, if you have a specific question about that, send me an email and I can send it on to the team. So are people not answering, or is it just, is it not working?
It said response recorded, but
Maybe you have
Only let
to
us
refresh or
Only let you select. Wow. Okay, it could be that it's all collecting. This is wireless from my computer to here, and so I don't know if that's how this.
the other thing was pretty.
The other one, yeah, it was a little. It was. Oh, we'll see. We'll see. It might all of a sudden explode. But send me an email, and
my question is very, very specific. Like whether there's plan to take those brains and maybe double the number of donors. 6 donors.
oh yeah,
Right.
yeah. No, I'll talk a little bit about that. But no, the model at the Allen Institute is somewhere between academia and industry. So the plan was to get 10 brains. It was never going to be about statistical significance. We could never collect enough data for that. But there's a couple things that it was hard to actually get normal brains. First of all, We have five male brains and one female brain online. I'm the one who gets all the emails at the Allen Institute, and we put the brains out serially. And so I got a lot of angry mail asking why there was not more female brains. And it turns out that the men tend to die in a particular way that allows for us to collect their brains, more of them. So there was a more frequency, which we knew, but it also turns out that men are less likely to donate their wives' brains than the other way around. So, and that one we had no idea about that one. So if you're female and you want to donate your brain, your body to science, let your family know. Yeah.
I'm curious, what's the particular way?
Well, so more heart attacks. You know, there's no blunt. You couldn't have any blunt trauma. So there was one of them died of an asthma attack, a really severe asthma attack. But so and there's.
Dying less violently.
Well, no, yeah, no. There was but there was actually way more male donors that we were able to choose from. There was actually a female brain that it looked normal past all the pathology and all this, and when they went to start cutting into it, they noticed she was missing a gyrus, an entire gyrus. So she was, and we told that to the family. They're like, oh. Right, but there was just that study that came out just a couple years ago, that 24-year-old woman who didn't have a cerebellum. So apparently missing a gyrus is no big deal, right? So
the problem is that the brains in the freezer basically do not meet certain standards.
Not necessarily. So we used her brain, but mostly as a way to standardize the procedure. There was a lot of, and I'll talk more about that data. I'm just gonna. It probably was collected, but
If you
it's
try
somewhere.
to back, just kind of refresh it. 'Cause I don't know.
Look at that! Yeah. Okay. Good. It's good to know.
Okay, better than just moving on.
Come on. Exactly. Okay. So this is. I want to talk to you about the data in the human brain atlas. So this is an image taken from our Nature paper in 2012, and there was a lot of processing that went into collecting the data. I'll walk you through some of the data some of the steps that we did to collect the data and how we collected it. But there was a lot of data that went into trying to figure out where we were in the brain. Mouse brain is really easy. All you got to do is zoom out and you can figure out where you are. Human brain, there's a lot of stuff that you have to do to figure out where you are. And then we store all of our data in a laboratory information management system, because there's just no way you could collect all that data in notebooks. There are no notebooks at the institute, and then there's a lot of analysis and informatics that goes on top of the data to produce a web resource that's available to you. So the first thing we did is we took a whole brain, and within 24 hours we got an MRI, either in cranio or ex cranio, depending on where we got it from. So they range from 18 to 68. That was the criteria. I think our youngest brain is 24. No known psychiatric or neuropathological history. No drugs, although everybody had theobromine and caffeine on board, so that's pretty normal. The MR scan was in Maryland, it was in the skull. At UCI in Irvine, it was done out of the skull. So we had to this is one of the processes that we had to work out. How do you get a brain without it being too smushed if you're going to do an MRI extracranially? And then the tissue was frozen, slabbed at half a centimeter to a centimeter, and that's how we received it at the Institute. And so the very first thing we did is we took a Nissl section, so that we could, of each block face, the first took a millimeter section, and this is actually a video just to show you what that looks like because it's a 6x8 slide. It's a huge slide, and so and then we stained it, and this way we could actually take a look at where we were at the beginning of that block face. And then we slabbed we took that slab and we cut it into blocks that would fit on 2 by 3. And there was a lot of histology we did, again, primarily to figure out where we were in the brain. You know, it's difficult to, you know, especially when you just have a chunk of brain, to figure out where you are. And it was done differently in the cortex and in the subcortex. The cortex, it was just a macro dissection. In the subcortex, we did micro dissection, laser micro dissection. And these are some images showing you, but I've also got, so there's a human brain. It's so cool to watch this. I know I could watch this one. But so there was a lot of histology that was done for each of these slides, and then on some of them we collected it on membranes so that you could actually. Isn't that cool? And then we have a whole system that does the ISH. So each of those are slides. And then the microdissection, we slightly Nissl stain the slides, and then we've got a tech who just draws on the section that they want to cut out. And you can see they cut out the section, it falls into a tube. All right, you can actually see the laser cutting right there. And then you extract the RNA and run it on a chip. Right, and so the data for a particular gene, for example, is put into the web, into our web resource. And how many of you have actually gone online and looked at the Human Brain Atlas web resource? So I want to walk you through that so that you understand our visualization as well as show you how to get the data. Yeah?
Sorry.
No, this is...
Very annoying, but...
No, I was about to say, this is very informal, and I will show you basically what you ask for, so this is good. So I appreciate any questions.
Okay, so can you go back a little bit, because
Yeah.
it wasn't quite sure. You say you do the microsections for the subcortical,
Subcortical and any...
and then you cut out the piece of cortex that goes into the...
Yeah, so if you look
And
at...
that's where you do the RNA.
Or extract the RNA, yeah.
But like 10 minutes ago, you said that you extract oh, sorry, that's the RNA, not the microRNA, or am I?
Well, no, this is right. This is
Same thing.
yeah. So that
Before you said it's a 1 by 1 by 1 millimeter cube.
that was yes. So if you look at here, this is from the final. You know, we slice a bunch of the brain, and then from the final millimeter, the back end of the brain, we macro dissect out all 6 layers of the cortex.
Okay.
So you see the little red squares on that one. So that, you know, we actually cut out different areas.
Okay.
Yeah, but then there's the weirdly shaped ones, right? Or the very small nuclei. Those were micro dissected out with laser.
Okay.
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 And then there's an extraction process that was actually different for each one of them because of how much tissue that was there. But and I'll show you the documentation so you can actually go in and look at that.
Right.
And so, did that answer your question?
It's probably going to get clearer later.
Well, yeah, but is there like...
They take the tissue, but then they micro dissect it from that, and that's what they use.
Okay. But it's still on a range of a millimeter, like you're not averaging over a big chunk of tissue.
No, yeah.
Very,
No,
very
it's
precise.
that's right.
The
So,
coordinate is less precise, but the tissue that they're doing is super.
yeah.
Yeah.
So,
Okay.
yeah, Cameron.
Did they, I mean, I guess I was trying to understand that, I'm sorry. Do you know what logic went into the imaging protocol they used?
For the MRI?
Yeah.
Well, mostly they had to get the brain frozen within 24 hours. And that was the logic. And they're
Just when they're doing 1 millimeter boxes, it was a great opportunity for you to get much finer resolution than that.
Right.
It's ex vivo. And I mean, something being frozen, isn't that bad for the MRI, is it?
well, it's actually frozen after the MRI.
Right.
So and it turns out that so we have DTI for some of the brains, but not for all of them, because it turned out that the DTI, it warmed up the brain too much for the RNA integrity. So it's and that was the whole thing, is we had to get it frozen fast so that you could still extract the RNA. Yeah.
Is the DTI online?
Yes.
In the maps?
I'll show you.
So you
Actually, we couldn't figure out a way to put that on our web app in a way that made sense or provided information for people. So it's available for you to download, but we haven't done anything with it. Okay, so I'll show you where to access that. Okay. Any other questions about that? Okay, so this is the data. This is the data that we have. So we've been collecting in situ hybridization data because that was our bread and butter at the beginning. So we've been collecting that the entire way. We have a subcortex study. It only looks at 55 genes across subcortical regions and one male, one female. We have a 1,000 gene survey. This was done on the top 1,000 genes that were searched for in the Mouse Brain Atlas. So it seemed to be the genes that were most of interest to people, and it was only in 2 areas of the cortex, the visual and the temporal cortex, and across multiple control brains. We have a schizophrenia study, just 60 genes. If you're interested in schizophrenia, in the dorsolateral prefrontal cortex, and in 50 controls and schizophrenic cases. So that's one of the disease studies that we have. We also, I know some of you are interested in autism, so we've got a small study, only again 25 genes. So literally all of the genes we have, it's about 1,000 genes because a lot of these are going to be repeated. And then for the Human Brain Atlas, we also did a neurotransmitter study. So there's about 176 genes across the brain that will be of interest, and they are taken from some of the same brains that we looked at the microarray. So we looked at a single hemisphere in 4 brains and 2 hemispheres in 2 brains. Those are the 6 brains. So we've got 8 hemispheres and 6 brains. We looked at approximately 500 samples per brain, and those samples were manually mapped back into the MRI space. And it was Agilent was who we decided to go to, and then all of the histology, MRI, and DTI data. So I'll show you how to access all of that data. Any questions about this? Okay, so we are known for the most part as the Google Maps of gene expression in the brain, and this is just to give you a range of how people use our data. This is one example of a study. They had a bunch of hypotheses about where in the brain their protein or their gene might be expressed, but they didn't know for sure because, again, when you're doing imaging analysis, it's hard to actually get a sample of the brain. But we have controls for that. So this was an example of using us like Google Maps. And then this student Ahmed. He completed his PhD without collecting a single bit of data. All of his dissertation was actually done on data from the Allen Institute, and there's 2 more graduate students in the pipeline, in fact. And this was his advisor last week. He says, "You keep doing what you're doing because your data analysis problem is our science, right?" They've created many different ways to access and look at the data. So this is his dissertation. My dissertation looked nothing like that. It was in this big binder, right, or binding. So I'll put up this presentation so that you can have access to this. But there's a lot of data analysis and tools that have used the Allen data. So anybody familiar with TS&E? Okay, cool. So this is the guy who actually developed it, and if you go to you can actually. He's got a lot of different links for you, different implementations. The Allen data, he links here to a paper. So, which is it's actually a very cool paper. But there's a lot of links here that you have access to. Again, this is another paper that links gene expression and imaging. And this was, several of you that participated in I'm asking actually now, the hackathon at the Human Brain Mapping meeting that was held here in Seattle in 2013. Was there anybody here involved in that? Excellent. Yes. So this was probably the first time in which people used neuroimagers put the gene expression data underneath it. So there's a lot of work that's been done since then, and I figure would be of use to people in this crowd. If you just do a search in GitHub for Allen data, there's a bunch of people who have done work with the Allen data. So.
Do you know who this guy is?
So which guy?
This guy who's asking all the questions.
Excellent. This is what's the hashtag?
Whoa, much called very brains.
That's right, exactly. So my favorite hashtag.
Can I add one more tool to this list?
Yes, absolutely.
So we just added, just a couple of months ago, gene decoding using Allen Brain Atlas.
Oh, excellent.
So it's a repository of statistical maps of the brain. It doesn't require any installation. You upload your map, you click a button, it decodes 20,000 genes.
Great.
Calculates.
Meet me right after this, and I'll put this in here before I upload this, so that people can click on this.
Go check it out. It's basically like Venga, but online.
Okay, excellent. Has anybody talked about neurosynth here at all? Say what? Yeah.
He's just took a phone call.
Is he? Okay, good. Yeah. So this is TALS. So so this is mapped into the gene expression space, the gene expression into the MNI space, and this one has to do with the semantics. So, and there was people talking about that as far as projects go. So, brain scanner, Bradley Wojtek. Anybody know? You've heard of them, obviously, right? So this is also, he also does this kind of thing where using associations between behavior and disease and the region of the brain, maps it onto the gene expression as well. So, like I said, there are many, many more. Christy's going to talk to you about one very soon. So, like I said, I'd like to get that one from you, Chris. So. Anybody interested in using the Allen Brain resources for your project this week? So I'm interested in knowing about it so I can provide the kind of resource that you might need this week. So let me know if somebody responds so that I can click back. Excellent. Now it's working in live time. Yes. Okay. Okay, great. Which one but not one of these? No, it's, who said E? Anyone? Yeah, who's in the mouse brain? Excellent. Okay, good. So, is that your 17%?
I can't be 17%.
Yes, so, okay, good.
Only 5 people.
Yeah.
So just the IV glioblastoma atlas,
Yeah.
how complete is that? Is it, you would only get little pieces, right?
Right. So we have a bunch of tumors, right? And we'll go into that data set because it's actually kind of complex because what they did a lot of they do an RNA-seq analysis and then take the top genes that showed up and do an ISH analysis on that. From that study would then do another RNA-seq analysis. So there's a lot of different data in that data set, and Jeremy will go into that tomorrow deeply so that you can actually get a sense of it. But it is one of the things you'll notice about our atlas is it has to do with it's either gene-centric or anatomy-centric. So that one's got the anatomy of of glioblastoma in there.
But not of the macroscopic brain?
Well, we do. You can access more data. So because we're a public database, we have to de-identify everything. But excuse me, you can access the Swedish. You just have to register on their site, and you can actually get the different meds that they were on, the treatment course that they were on, like that. So there's a lot of extra data, including a lot of imaging. Yeah. So we'll show you how to access that tomorrow, or today if you want to jump in right now. Okay, great. Somebody else? Did somebody else? Oh no, 13% is probably just the mouse data. Okay, good. Okay, so there's a couple things that, so you're welcome to jump on brain-map.org right now, and because there's a couple things that I want to show you, regardless of what product that you go into, there's specific things that you're going to need to know. So the first is there's a search box in the top. If you've got a question about anything, that's a good first place to start. It'll take you to a faceted search results. Basically, it shows you all of the experiments that have been done on your particular search. There's an overview page. So this overview page is a brief blurb on all of the different resources, including links to tutorials, to documentation, to the online help, things like that. There's also tutorials. So these are short videos, 5 to 7 minute videos that'll show you how to get in and out of a particular resource. Documentation. Especially if you're going to publish on the data, I highly recommend you read how you collected it. So and if you have any questions about anything that we've done, it's going to be in there. And of course, if it's not in there, you can always send me an email and I'll go hunt down the scientist and figure out what exactly happened. There's also online help. So if you know that you should be able to do something, but you can't figure out how to do it, this is a great first place to start. And then there's also our reference atlases. So this is this tool includes both the mouse brain atlases, developmental as well as adult, as well as our human atlases. So we have a 34-year-old human as a reference brain, as well as 2 fetal atlases as well, 15 post-conception week and 21 post-conception week. Okay, so this is a piece of work that was just recently published, but this is a figure that you have access to from our website. It's the genetic geography of the brain on our, if you look on the front page, and this looks at gene expression across all 6 brains. So, and what can that kind of data tell you? And so what we've got here is we've got all the different structures plotted against all the different structures here. And what you're looking at in this graph is the differential expression. So how different is the gene expression from one region to another? So for example, notice that when you compare one structure to another structure, you get the light blue. So blue is no differential expression, and then the red is highly differentially expressed. So there's, just to give you a sense of it, this is the hippocampus. So you'll notice that in each of the different regions of the hippocampus, it's actually differentially expressed all the way across. And if you know anything about the hippocampus, you know that each of the regions shows very different gene expression. And also, this is the cortex. You can also see over here, this is the cerebellum, the old cortex, and this is the new cortex, the neocortex. And you'll notice that it's primarily blue, only blue in the cortex here, in the cerebellum here. But what that says to us is that, and remember, when we took samples from the human brain, we took all 6 layers of the cortex. So this isn't laminar expression, because we know that gene expression changes over the lamina. But when you're looking at gene expression across the whole cortex, there's not much change. So the front of the cortex looks very similar to the back of the cortex, and that actually was the basis of a lot of our hypotheses, that there's a functional unit. It's not only our hypotheses, but there's a functional unit in the cortex. And so if we can figure out how that part of the cortex works, it should be able to translate to other regions of the cortex as well. So, and then this little cross that you're seeing in here, this is the visual system. So it is the one area in the cortex that we're studying now that is a little bit more differentially expressed, but it's still very different. It's similar to it itself compared to the rest of the brain. Okay, so what I want to do is I'm just going to get out of these guys. Oh, I don't know if I want to log out of that, so I'm not going to. No, that's not what I wanted. That's my budget. Okay, so there's a couple ways to get at everything here. If you go to this link, it'll take you to, there's a bit of a delay there, so I'm going to go from here. Okay, so what I want to do is I want to show you the web interface, because you could use this to look at the data that you're downloading, especially if you're going to download it, in big chunks. So we've got if you don't know any genes, right? Some of you might not have a favorite gene, right? Hard to believe, but it's possible. But you can right now either type in your favorite gene, or I'm using prodynorphin. I used to work in the opioids. And I'm going to set this up as you're probably seeing this. Okay, so what I'd like to do is just walk you through a little bit of what you're seeing so that you know how to access everything that you need to access. So what you're looking at here is the list of probes. So for the most part, every single gene has 2 probes. And prodynorphin, we tiled the probes across the gene. So we've got 14 different probes here, 14 or 16. We've got 2 column headers that are color-coded here. The top one is the donor. You've got this, you see the 6 different donors, and then underneath that you have the different structures. So you can sort by structure instead of sorting by donor. So you'll notice now all the brains are interleaved, and you've got the different structures here. And what you're looking at, the data, is a z-score. So each probe, we normalize the data across all the structures, across all 6 brains. So what you're looking at is average gene expression in the black, underexpressed in the green, overexpressed in the red. So the z-score is solely for visualization purposes. You actually don't want to download the z-score, because it basically just gives you, it's a way to identify areas of the brain. So for example, prodynorphin is high in the striatum. You can see this red stripe here. It's high in different areas of the amygdala. It's high in the dentate gyrus. So you can kind of get a sense. It actually draws your eye to particular regions. The red-green is not good if you're colorblind, or it doesn't usually project very well. We've got a different color. But if you're going to download the data from the web resource, you want to do the log2 scale. This is the normalized data, and you can see here that the different probes have different binding affinities. This one obviously binds tighter than, say, this one, but it's all very similar, which comes out in the z-score. Yeah.
Do you have any advice on choosing or averaging over probes?
Yeah, there's a method that we used, especially for the big one. So if you've got 1 probe, obviously use that one. If you've got 2, use the one that's the highest. And if there's more,
That's the strongest expression.
Yes.
Across all.
Yeah.
Okay.
And then if you've got 3, do a correlation and see and choose the one that has the highest correlation. All of them. So compare the correlation of each probe to each probe. So there's. Yeah. Does that? I can actually show you. So I'm actually going to go, like I said, I'm going to show you guys what you ask for, right? So how many of you are familiar with the API? Okay, good. So all of our data is available from our application programming interface, and I'm going to just do this so I can save that. So all of our data is available from here. There's an examples page here. And let's just go straight into the human. So this is an example of how you can find the correlation between each of the different probes. And you've got this either. I'm not a programmer, so I'm just pointing you to this. Hopefully you can access it and do way better at it than I can. So, but this is an example of different different visualizations that we didn't use in our website, but our developers messed around with. So we put them someplace so you have access to them if you'd like.
That second example, the SVM activity expression correlation, is the prototypical first piece of code doing that imaging.
Probably, right. Yeah, not that I would know.
The very first one that it's ever put out there.
Oh, for this data? Oh, yeah.
Or
Yes.
brain scanner.
Yep. Yeah. So these are some of the examples, and then also we have our software development kit. This is basically and right now it looks at these different data sets. So it doesn't have the other data sets in there, nor does it have the CCF, although the CCF, the Common Coordinate Framework, is going to go in come October. Okay, and then after we finish up our mouse Common Coordinate Framework in sometime next year, we're going to work on our human Common Coordinate Framework as well. Okay, so yay Python. I just started learning Python a couple months ago so I can answer questions on the web. So, okay, so any questions about what you're looking at here? Okay, the page that's probably going to be of most use to you is going to be this download page and the MRI page. So if you click on the MRI page, we've got 8 different brains that you can download. So we've got the T1 and the T2. Again, DTI only for a few of them. So, and the 2001 and 2002 were the in-cranial brains. And then the rest of them were done ex-cranial. Okay, and then, yes?
I was wondering, so the extracranial brains, they made the holders for them? Mm-hmm. Did they use did they customize the holder for each brain? Or
yes.
Did they use a single?
Well, I think because it was in kind of a putty plastic thing that they put it inside. So.
Because if you look at the brains, go back. If you look, going back to what you just did a second ago.
Yeah.
I don't know how useful this is, but so if you look at that one and you compare it to the one next to it, overall. So some of them look like they have much similar brain shape. Yeah. And some look very different. So either they were somehow normalized to one another, or maybe like how the holder was kind of. So actually, go down further. Yeah. The ones that are in the.
Yes, these ones.
See how the brain shape varies much more between individuals than the ones that were.
Yeah, big. They're bigger up at this end, and also the contrast is kind of wonky at the front and back ends as well.
You know, I was just wondering if the container they had kind of a shape on it.
Well, this one looks kind of.
Anyway.
Yeah, and it's, if you go through it and look at the documentation, it talks about that a bit. So, that they are, we didn't create an average brain because we have 6 brains, right? So that's, yeah.
Just as a random comment on that, I actually used these subjects to do some brain morphology analyses in a paper earlier this year. It was more of a follow-up to show that, at least with older adults, people tend to move more in the scanner. So these are older adults that are not going to move in the scanner, at least across the span. So to show, not only statistically, but just is there any trend in that direction? And things made sense.
Oh,
so
cool.
I didn't look as closely to see how they were for the ex vivo, out of the cranial ones, how that was done. But the results did make sense still.
Cool. I'd like to, if you could send me a link to that paper, that'd be great. Okay, and then from the download. So probably how you're best going to want to download this data is from this page. So this is going to give you a giant matrix of all of the expression data. But one of the things that goes along with it is the MNI coordinates. It's got the MR coordinates, but then there's a transform that's it's the same for every brain. I mean, it's different for every brain. So you'll have the MNI transform already calculated for you, so you can. And I've been told that that means that some of the points are outside the brain, but it's for the most part it really works. When you download the data directly from the, let's see, when you download the data directly from the website, which you also can do if you want to get a smaller set, you're only going to get the MR coordinates because it's all 6 brains. And then, but how you do that, if you're interested in that, you can actually make this, because this is a huge amount of data, right? But typically, especially people at a hackathon want all of the data, but if you want a smaller subset of the data, you can use this feature. So you can actually just select particular donors. You can also select particular structures. And when you do that and turn the heat map filters on, right, you can actually just download this little piece of data, this little section of data as well. So if you're only interested in a particular gene, notice also the resolution. We've got structures and samples. You probably want to download the sample. There are regions of the brain that will be more oversampled than other regions of the brain. And so when we put it on the web, it's in structures by default. So we rolled up the gene expression in one area to that structure. Okay, and then you can also. So this is I put on Slack this particular atlas, or this viewer. This is the Brain Explorer. Like I said, we didn't create an average brain, so we have all 6 MRIs, the MRI space. And what you're looking at, the dots are the subcortical regions, and you've also got the cortical regions as well. So this is an inflated cortical surface, and this, since it wouldn't make sense to do just a single point from which we extracted the DNA from, or the RNA from, it's kind of splayed on the gene expression is actually splayed on that area. Some of these you can actually.
In reality, it was just a tiny millimeter size piece of the cortex. Inside
that area.
In this visualization, you assume that the gene expression is homogeneous over a much bigger region.
Exactly.
How did you pick those boundaries?
Right.
You're gonna look at.
So you've got I'm sampling from point 1 and sampling from point 2.
Right.
But then you inflate
it, how big it was,
how big it is, and where the boundaries
and where the boundaries were.
Yeah.
It was all done by our data viz guy. If it's not in the documentation, right? Okay. I couldn't tell you myself, but I can point you to where to find it, which is inside this documentation. Okay. So.
Some sort of parcellation.
Yeah, definitely.
Whatever they started on.
Yeah.
So it may very well be a standardized.
Here. You can look. So yeah.
It's not the Craddock 400.
So and you can actually see the M&I coordinates here. So it's obviously a smaller space inside of this. Okay, and then if you're interested in the Brain Explorer, I could spend plenty of time just walking through how to use the Brain Explorer as well. Okay, so that's one of the ways to visualize the data. And if you're, for example, let's turn this off. And you actually want to go drill down into the data. If you select the heat map, we've got each of the samples where they were taken from overlaid on the MRI, and we've got the heat map here as well as. Oh, this is still buggy. This is going to get fixed. It broke in the last release, so you can see that the SVG images are off from the. But we have where that spot was actually taken from in these. And it's interactive as well. So when you click on the heat map, it'll take you to that spot in the MRI. It'll also take you all the way down to where it was taken from. And then there's a couple different things here. So you can, if you're interested in a particular region and you want to know what the gene expression in that region is, it's the differential search that you're interested in. So in the differential search, you can actually type in different structures, a structure or different structures, and then a contrast region. So what it's going to look for is genes that show enhanced expression compared to the gene expression in that contrast region. And you can do that, so you can look at the dentate gyrus inside of the hippocampus, or you can look at the dentate gyrus inside of the entire brain, or you can look in the, the hippocampus versus the thalamus, right? So it's that kind of a search. And then also, once you find a gene that shows an interesting expression pattern, you can also find genes that show a similar expression pattern using the correlate search. So what got returned is all the genes that show a similar expression pattern to protonorphin, and protonorphin is one of the top choices, which is good. But you can actually look down and see other genes that show a similar expression pattern. So those are some of the kinds of searches that you can do. So if you're looking for a set of genes that shows similar properties, these are the different kinds of searches that you can use. Okay, any questions about accessing any of this data? This is just scratching the surface. So again, check out the documentation. And then if you find, for example, this is protonorphin expression in a chunk of tissue, right? So there's a lot of different cells involved in that. One of the things that you can do is look to see if we've got any in situ hybridization data for the human. Now, that's cellular resolution data, so it's possible that you might be able to see something inside the morphology of the cells itself, but you have to check to make sure that we actually have that data, first of all. So if you look, we have 209 experiments for protonorphin, and you can actually see that we've got it in humans. So this is actually the best way to look. So if you're looking for in situ hybridization data, this is the best way to look. Find the gene that's of interest to you first, and then you can go look at it. Oh, you can't see it from... There's actually data on the screen here. It's very high resolution, and it's actually pretty highly expressed there. So the data is actually very high resolution, and you're welcome to download the data. That if you're... one of the interesting things about humans is you can't just zoom out and figure out where you are. So this is one of the ways that we give you access to where you are in the brain. So we've got hotspots here. If you click on one of the hotspots, or if you click, I can show you, it'll actually take you to the reference brain. So sometimes I get used to seeing the mouse brain and then trying to figure out that region in the human brain. Sometimes it makes sense to just go and look at the context to be able to see whereabouts you are in the human brain. So I jumped all over there. Probably where it's going to be of most interest to you is downloading the data. Okay, so any questions about anything? Because what I want to do now is I want to turn it over to Kirsty. Sure. Right.